we are looking at 1.1.NOS1. NOS stands for Nature of Science, in case you were curious. So let's jump in. Today's lecture is on trends and discrepancies. So although most organisms do conform to the cell theory, there are exceptions. Any rule has exceptions. So let's jump right in. And this goes with your notes as well. All right, so what is a trend and what is a discrepancy? A trend is a prevailing tendency, a generalization, okay? It's something that tends to be true. Get it? Tendency. A trend is something that tends to be true. A discrepancy is something that does not fit the general rule, is an exception, all right? It doesn't fit the trend. So trends lead to development of predictions in what we observe. Here's the thing. You have to have trends to move forward. So you cannot make predictions if there are not trends, right? So all animal cells, they tend to behave in a certain way, depending on the grouping, right? And so these cells make these trends, and because of that, you can make predictions on how they will behave, what you should see. If I say I'm going to look at an animal cell under a microscope, I know what I should see. I know I should see a nucleus, right? If it's an animal cell, I should see a nucleus. I know I shouldn't see any cell walls. I should see a rough and smooth endoplasmic reticulum. I should see a Golgi apparatus, some ribosomes, lysosomes, mitochondria, probably some vacuoles. There are things that you expect to see. And these expectations are based off of the trends, these general rules of thumb that you have grown accustomed to that are true for the masses, all right? But a discrepancies pop up in data all the time and are just as interesting as the trend. So the trends allow you to make predictions. Discrepancies are important to note. Is there something that caused that discrepancy? Is there a way to manipulate a situation so that it always causes that discrepancy? And overall, it gives you just this much deeper understanding of how things work. So let's keep going. The examples are key. Oh wait, before I do that, go back. Discrepancies in statistics are particularly significant in some circumstances and absolutely insignificant in others. Sometimes it's a human error. So when we get to our internal assessments, our IAs, keep all of this in mind that you should have trends and discrepancies. Okay. Aw, baby's cute. Okay, so list features that would be considered a trend related to the cell theory. So we have three main ones, right? We have all living things are composed entirely of true cells. Two, a trend is that cells are small in size, right? You don't ever think of a cell as something like this big. And there are trends in typical cell structures such as, and those would be membrane-bound organelles and eukaryotes, which we just spoke about. This is interesting. So if you look down here at sizes of typical cells, we are in micrometers. So I love that they included this conversion table over here. So if a centimeter is 10 to the negative second meters, then we go down here to a micrometer and it's 10 to the negative 6. So we're talking so small, so small. And that's the size of most eukaryotic plant and animal cells are in micrometers. But you get down to a virus, and viruses are in nanometers. So they are vastly smaller than what you can imagine a cell being. So anyway, cells are typically really small. Okay, so in a cell, some trends that you typically see, we've talked about membrane-bound organelles and eukaryotes. Genetic material should be found in some way. Energy should be released, metabolism. It should be formed in our bodies. That's mitochondria. They should create proteins. Cytoplasm should have enzymes to help with metabolism and speed it up. And then there should be a cell membrane. Every living thing has a cell membrane. At least that's the trend. All right, let's go on. All right, let's talk about discrepancies. I'm trying to get my face out of the way. Okay, so discrepancies, giant algae. They are huge in comparison, huge. So here's a little picture off to the right. They are a very large single-celled organism. Normally you need a microscope. If you can see in the picture, that's a person's hand. You can hold these in your hand and turn them about. Anyway, crazy. So that's a discrepancy. Mammal skeletal muscle cells. So our muscle cells have more than one nuclei. Not only are they large, but they're multinucleated. And that's so weird. Think about it. What other cells have more than one nucleus in them that you know? Right? So these muscle cells have more than one nucleus in them, and they're much larger than your average cell. They are not necessarily always comparable to the giant algae, but they're much larger than standard. All right, aseptate. So aseptate, aseptal, fungal hyphae. This is interesting because they're not actually in defined cells. They're not clearly defined as cells. They sort of just look like branches, and then they behave. But there's no clear division of a cell. They all just kind of smear and branch. And so they're all over the place. It's very different than when you think of a perfect circle or little cubes as you do with most cells. Okay, so let's talk about discrepancies for a second. We are going to start, and this is all still in your packet, with red blood cells. This is crazy. You need blood to be alive as a human being and as an animal. They're called erythrocytes. That's the red blood cell, right? Leukocytes are white blood cells. And the whole point is to carry oxygen and hemoglobin throughout your body, right? It's taking oxygen to those tissues to keep those tissues alive. Now, here's the crazy part. During the maturing process, during the maturation, they actually toss out their nucleus and their mitochondria. They don't have either. So that's the discrepancy. They don't have a nucleus and they don't have a mitochondria. That's crazy. How do they even power themselves? How do they have genetic information? Well, once they're mature, theoretically, your DNA would not be in your blood cells. So if all someone had of yours was just your red blood cells only, they shouldn't be able to tell that it's your blood. And thus, there's a whole other conundrum of issues that come with that. Also, cool articles down there. More discrepancies. Let's see here. What else do I have for you? I have, vitreous humor. What is vitreous humor? Vitreous humor is the fluid behind the lens that keeps the retina in place. So normal bone and osteoporosis. Osteoporosis is far more common in women than in men. It has to do with the female body and how the female body deals with calcium and hormones and all of that. The males can have osteoporosis. They just tend to deal with it differently. Their body and calcium deals differently. So crystallized mineral deposits in the bone, and they provide strength and rigidity for the bone matrix. So your bone marrow and the actual substance of your bone is porous, but it needs to be like spongy, but also rigid because it has to hold you up, right? It has to like protect you, right? Like that has to be kind of hard, but in the inside, it has to still be spongy. So anyway, just some other things that don't necessarily go with the flow. Okay, and then xylem tissue and vascular plants. This is how the food and such gets up the tubes. It works like little straws. You have the xylem and the phloem. And let me think. Xylem is for water, so xylem is more like a drinking straw. But either way, these cells do not look or act in a way that other cells do. What else? Let's see here. Some things to add. Your bone mineral deposits, they do provide strength and rigidity for the bone matrix. Xylem tissues, the interesting thing about xylem tissues is that they are made when cells actually die. So there's sort of just gaps when cells die, and they leave the walls as these hollow tubes. They do provide support and the path for water conduction, but they're there because something died, not because something was made. So anyway, that's really, really interesting. And then here, of course, is more things to discuss about trends and discrepancies and other chances where we will probably talk some more about these topics. Okay, until next time, bye.